Autism spectrum disorder or ASD, commonly known simply as autism, is a neurodevelopmental disorder characterized by difficulties in social interaction, verbal and nonverbal communication, and the presence of repetitive behavior and restricted interests. Other common symptoms include unusual responses to sensory stimuli, and an insistence on sameness or strict adherence to routine. As per WHO estimates, 1 in 100 children around the world have autism of various intensities. Even though many homeopaths produce excellent results in alleviating troublesome symptoms in autistic persons using prescriptions made on the basis of totality of mental and physical symptoms, complete cure is very rare. This fact points to the need for a more comprehensive and scientific approach. MIT homeopathic prescriptions based on deeper study of pathophysiology of autism, as well as a rational understanding of underlying miasmatic background, inevitably related with the off-target actions of antibodies generated against alien proteins such as infectious agents. ASD includes a wide variety of characteristics. Some of these include behavioral characteristics which widely range from slow development of social and learning skills to difficulties in creating connections with other people. Other behavioral characteristics include abnormal responses to sensations such as sights, sounds, touch, taste and smell, and problems of consistent speech rhythm. The latter problem influences an individual's social skills, leading to potential problems in how they are understood by communication partners. In order to be diagnosed with ASD, a person must have at least 2 of the following behaviors. 1. Repetitive behaviors such as rocking, hand flapping, finger flicking, head banging, or repeating phrases or sounds. These behaviors may occur constantly or only when the person gets stressed, anxious or upset. These behaviors are also known as stimming. 2. Resistance to change. A strict adherence to routines such as eating certain foods in a specific order, or taking the same path to school every day. The individual may become distressed if there is any change or disruption to their routine. 3. Restricted interests and excessive interest in a particular activity, topic, or hobby, and devoting all their attention to it. For example, young children might completely focus on things that spin and ignore everything else. Older children might try to learn everything about a single topic, such as the weather or sports, and persevere at or talk about it constantly. 4. Sensory reactivity. An unusual reaction to certain sensory inputs such as having a negative reaction to specific sounds or textures, being fascinated by lights or movements or having an apparent indifference to pain or heat. Autistic individuals can display many forms of repetitive or restricted behavior, such as stereotype behaviors, compulsive behaviors, ritualistic behavior, self-injurious behaviors etc. Self-injury could be a response to modulate pain perception when chronic pain or other health problems that cause pain are present. An abnormal basal ganglia connectivity may predispose to self-injurious behavior. Autistic individuals may have symptoms that do not contribute to the official diagnosis, but that can affect the individual or the family. Some individuals with ASD show unusual abilities, ranging from splinter skills such as memorization.
There are studies describing how some individuals with ASD have superior skills in perception and attention, relative to the general population. Unusual eating behavior occurs in about 3/4 of children with ASD, to the extent that it was formerly considered as a diagnostic indicator. Selectivity is the most common problem, although eating rituals and food refusal also occur. Gastrointestinal problems are one of the most commonly co-occurring medical conditions in autistic people. These are linked to greater social impairment, irritability, language impairments, mood changes, and behavior and sleep problems. Pathological demand avoidance can occur. People with this set of autistic symptoms are more likely to refuse to do what is asked or expected of them, even to activities they enjoy. The most common medical conditions occurring in individuals with ASDs are seizure disorder or epilepsy, tuberous sclerosis, an autosomal dominant genetic condition in which non-malignant tumors grow in the brain and on other vital organs, intellectual disabilities, learning disabilities, various anxiety disorders, schizophrenia, behavior problems, attention deficit hyperactivity disorder ADHD, sensory processing disorder, schizoid personality disorder etc. Several metabolic defects, such as phenylketonuria, are associated with autistic symptoms. Sleep problems affect about 2/3 of individuals with ASD at some point in childhood. These most commonly include symptoms of insomnia such as difficulty in falling asleep, frequent nocturnal awakenings, and early morning awakenings. Sleep problems are associated with difficult behaviors and family stress, and are often a focus of clinical attention over and above the primary ASD diagnosis.
Autism spectrum disorders are considered to affect information processing in the brain by altering how nerve cells and their synapses connect and organize, but the exact causes and biological mechanism of this process is not so far well understood. Even though autism has a strong genetic basis, it is not an inherited genetic disease. It is not associated with any inherited chromosome abnormality. Rather, it is the mutational outcome of epigenetic factors influencing the neuron development during the early stages of fetal development and postnatal period. Many factors known to be teratogens that cause birth defects are also associated with development of autism. Environmental causes, drugs, heavy metals, pesticides, maternal and childhood vaccines, maternal and prenatal infections, emotional shocks in mothers, alcoholism, illicit drugs, smoking. There are a lot of factors belonging to epigenetic spectrum of causes that lead to autism in a child. Certain foods, solvents, diesel exhaust, pH phthalates and phenols used in plastic products, pesticides, insect repellents, brominated flame retardants are also supposed to be implicated. Any chemical molecule that may bind to enzymes involved in biomolecular processes of genetic expressions associated with development of neurons and synapses during prenatal and postnatal phases of infants may lead to development of autism. Radiations that are composed of particles that carry enough kinetic energy to liberate an electron from an atom or molecule, can produce changes in various biological molecules and liberate hyperactive superoxides. These superoxide radicals bind to enzymes associated with genetic expression, chromosome folding, histone methylation etc., leading to various epigenetic errors. If these epigenetic errors happen during developmental stages of neurons and synapses, it may lead to autism. The presence of antibodies directed against components of the CNS in the sera of autistic children is indicative of an immune process that may be involved in the pathology of some cases of ASD. Autoimmunity develops when the immune system is inappropriately directed to recognize and exert an exaggerated response to self-components. The exact mechanism of autoimmunity in autoimmune diseases is not identical, but they all have autoreactive antibodies and T cells. Almost with all autoimmune diseases, genetic, immune and environmental factors, such as diet, toxic chemicals and infections, play critical roles in the development of the disease. Casein, caseomorphins, gluten and gluteomorphins, the opioid peptides, which is present in a range of food sources, are all implicated in the etiology of autism spectrum disorders. These peptides can stimulate T cells, induce peptide-specific T cell responses, and can lead to abnormal levels of cytokine production, which may result in inflammation, autoimmune reactions and disruption of neuroimmune communications. In autism spectrum disorders, majority of children have wheat and milk protein intolerance. And accordingly, removal of these peptides from the diet significantly improves their conditions. Immunoglobulins, such as IgG, IgM and IgA, were detected against 9 specific neuron-specific antigens in the sera of children with autism. These antibodies were found to bind with different central nervous system molecules that have sequence homologies to a milk protein called casein. This phenomenon points to the role of antibodies generated against milk protein casein in the causation of autism, by binding to different central nervous system molecules. Emerging studies indicate possible associations between viral infections and neurodegenerative and neurobehavioral conditions including autism. Viral infection during critical periods of early in utero neurodevelopment may lead to increased risk of autism in the offspring. Many infectious agents including measles, rubella virus and cytomegalovirus vaccines have long been suggested as etiologic factors in autism. A complex communication system does exist between the nervous and the immune system, during normal and pathological conditions. Alteration in brain function can result from immune cells and molecules, such as cytokines and chemokines. This might affect cognition and emotions. Furthermore, immune cells and immune molecules can result in neuronal modulation of systemic CNS responses to infection, injury, and inflammation. The cytokines have been shown to directly affect neural tissue function and development, especially the pro-inflammatory cytokines. Cytokines have been suggested to be responsible for many common features of autism, such as mood and sleep disturbances. In turn, neuropeptides, derived from the central and peripheral nervous system, have profound effects on the immune system, including the chemotaxis and recruitment of innate immune cells. Dysregulated immune function has been suggested by many scientists. Systemic, immunologic abnormalities in autism have been related to the generation of antibodies that are reactive to CNS proteins and have the potential for neuronal tissue destruction, or leading to an inappropriate or ineffective immune response to pathogen assault. Maternal immune response to infection can influence fetal brain development via increased levels of circulating cytokines. Autoimmunity was first linked to autism in a study of an autistic child, with strong family history of autoimmune diseases. This study suggested that an inherited risk of autoimmunity could increase the risk of developing autism. Another study showed the mean number of autoimmune disorders to be greater in families with autism. In most of these cases, the individual with the autoimmune disorder was a first-degree relative. A variety of anti-brain antibodies have been found in autistic patients, including antibodies attacking serotonin receptor, myelin basic protein MBP, neuron axon filament protein, cerebellar neurofilaments, nerve growth factor, 2-adrenergic, adrenergic binding sites, anti-brain endothelial cell proteins, and antibodies directed against an as yet unknown brain protein. It was found that mothers diagnosed with asthma or allergies during their second trimester were more than twice as likely to have a child with autism. This fact also points to the role of antibodies as the causative factors of autism. Despite that the fact there is increase in ASD research worldwide, the exact etiology of autism and ASD remains largely unknown. Over the last few years, a scientific interest has occurred in the close relationship of the immune system to the central nervous system, leading to considerable expansion in the field of psychoneuroimmunology. And currently it is widely accepted that environmental factors can compromise the immune system. There is emerging evidence and growing concern that a dysregulated or abnormal immune responses play an important role in some forms of ASD. In general, the associations between the immune and neurological systems are becoming more evident in many neurological disorders. Behaviors such as mood and sleep can be altered by cytokines and other products of immune activation, due to widespread effects on neurons. Aberrant immune activity during the early critical periods of brain and neuronal development could potentially play a role in neuronal dysfunction. Several efforts have attempted to link dysfunctional immune activity and ASD, such as maternal immune abnormalities during early pregnancy, increased incidence of familial autoimmunity, and childhood vaccinations. Several lines of research have shown abnormalities of the immune response in autism, including abnormal generation of antibodies, cytokines, and immune cells. Furthermore, In recent studies, antibodies directed against the fetal brain have been detected in some mothers of children with autism. These antibodies have the ability to alter behavioral outcomes in the offspring of animal models. A better understanding of the involvement of the immune response in early brain development, with respect to autism, may have important therapeutic implications. Although the specific etiology of ASD remain unknown, many hypotheses regarding causation of ASD abound, including the potential involvement of the immune system. A significant amount of research linking ASD and aberrant immune function has taken place, and although inconsistencies still exist, a clearer picture of the importance of immune involvement in ASD has emerged. Studies have shown an increased prevalence of so-called familial autoimmune disorders in ASD. A study found that 46 of ASD families had 2 or more members with autoimmune disorders, and as the number of autoimmune disorders within the family rose from 1 to 3, the odds ratio for a risk of a child with ASD increased from 1.9 to 5.5. This study found that autoimmune prevalence was significantly increased in mothers and first-degree family members of ASD subjects, with type 1 diabetes T1DM, rheumatoid arthritis RA, hypothyroid and systemic lupus erythematosus SLE being the most common disorders found. One study also found a specific link to hypothyroid Hashimoto's thyroiditis and RA in parents with ASD offspring. Recent reports have suggested a tight relationship between viral infections and neurodevelopmental disorders. In this regard, fetal brain damage can be caused by antibodies generated against viral infection or induced immune responses and cytokine storm. Epidemiological data suggest that viral infections during pregnancy are associated with high risk of having an autistic child. ASD is programmed in the mother's womb and it is the uterine, not peripheral, immune activation that works as the initial trigger to induce fetal brain developmental anomalies. Obstetric infections are particularly dangerous to both the mother and the developing fetus as they may orchestrate events that interfere with normal fetal developmental programs. The placenta is now considered a specialized immune organ, and in this regard, intrauterine infections may adversely affect the immune balance regulated by the placenta in concert with the maternal immune system. Viruses have evolved contemporary ways to evade the immune system and to cause diseases. Local cytokine storm and functionally or proportionally altered immune cell profiles have been the main consequences of viral infections. Other co-infections by bacteria or parasites may further compound these immune responses. In the case of fetal growth and survival, these infections will have enormous deleterious effects. The well-known congenitally acquired infections pathogens that cause morbidity and mortality in newborns are Toxoplasma gondii, Treponema pallidum, rubella, cytomegalovirus, and herpes simplex virus infections. This group has been expanded to include parvovirus B19, human immunodeficiency virus HIV, varicella zoster virus, hepatitis C virus HCV, Zika virus ZIKV, and Plasmodium falciparum among others. These maternal infections are passed either transplacentally or during the birth process. These pathogens may act independently or in concert to cause neonatal morbidities. More importantly, an array of literature now suggests an association between viral infections and poor fetal brain development and childhood diseases such as autism, schizophrenia, bipolar disorders, microcephaly and other serious brain disorders. As a matter of fact, several of these adverse neurobehavioral outcomes share the perinatal inflammation pathogenesis. Genetic causes have a crucial role in the development of ASD-like disorders, but early exposure to environmental factors during brain development has been shown to significantly increase the severity of the disorder. Since inflammation, cytokine imbalance, and viral neurotropism could have an impact on fetal brain development, the potential role of immunological dysregulation in autism has garnered particular attention. Viruses may cause ASD by directly infecting the brain, by inducing local and/or systemic cytokine storm, or by altering maternal or offspring localized immune responses. Several prior studies have linked ASD to various viral infections. Local cytokine storm and functionally or proportionally altered immune cell profiles have been the main consequences of viral infections. Other co-infections by bacteria or parasites may further compound these immune responses. In the case of fetal growth and survival, these infections will have enormous deleterious effects. The well-known congenitally acquired infections pathogens that cause morbidity and mortality in newborns are Toxoplasma gondii, Treponema pallidum, rubella, cytomegalovirus, and herpes simplex virus infections. This group has been expanded to include parvovirus B19, human immunodeficiency virus HIV, varicella zoster virus, hepatitis C virus HCV, Zika virus ZIKV, and Plasmodium falciparum among others. These maternal infections are passed either transplacentally or during the birth process. These pathogens may act independently or in concert to cause neonatal morbidities. More importantly, an array of literature now suggests an association between viral infections and poor fetal brain development and childhood diseases such as autism, schizophrenia, bipolar disorders, microcephaly and other serious brain disorders. As a matter of fact, several of these adverse neurobehavioral outcomes share the perinatal inflammation pathogenesis. Genetic causes have a crucial role in the development of ASD-like disorders, but early exposure to environmental factors during brain development has been shown to significantly increase the severity of the disorder. Since inflammation, cytokine imbalance, and viral neurotropism could have an impact on fetal brain development, the potential role of immunological dysregulation in autism has garnered particular attention. Viruses may cause ASD by directly infecting the brain, by inducing local and/or systemic cytokine storm, or by altering maternal or offspring localized immune responses. Several prior studies have linked ASD to various viral infections.
How viruses can access cellular components of the placenta and the pathogenic mechanisms that facilitate the process of specialized inflammation at the maternal-fetal interface is a subject of intense discussion. Maternal infections during pregnancy are associated with intellectual disability and autism in exposed children. Whether these associations are causal, and therefore should be targets of preventive strategies, remains unknown. We aim to investigate these associations, to determine whether there is a causal role of maternal infection during pregnancy for children's risk of autism and intellectual disability, by accounting for unmeasured familial factors. New clues hint that young boys who get serious viral infections might be more likely to develop autism. Scientists may have found a link between serious childhood infections and autism later in life. Scientists very close to understanding what causes autism and how to treat it. A study has already proved that severe infections in childhood might make a future diagnosis of autism spectrum disorder more likely in men who are genetically predisposed to the condition. While genetic factors are understood to be a key driver of autism, the new study bolsters the idea that genes don't necessarily trigger autism on their own. Environmental factors, like a viral infection, also play a role. The immune responses generated by organisms that cause perinatal maternal infection, i.e., bacteria, viruses, or parasites, have been associated with the development of autism in offspring. There is currently an increasing suspicion among researchers that autism is a complex disorder whose core aspects have distinct causes that often co-occur. While it is unlikely that a single cause for ASD exists, many risk factors may contribute to ASD development. These risk factors include genetics, prenatal and perinatal factors, neuroanatomical abnormalities, and environmental factors. It is possible to identify general factors, but much more difficult to pinpoint specific factors. Given the current state of knowledge, prediction can only be of a global nature and therefore requires the use of general markers. Coronavirus primarily targets the human respiratory system, but it also has neuro-invasive capabilities by spreading from the respiratory tract to the CNS. Upon nasal infection, The virus enters the CNS through the olfactory bulb and can eventually lead to inflammation and demyelination. The neuroinvasive abilities of the coronavirus was evidenced by COVID-19 patients presenting with febrile seizures, encephalitis, convulsions, and altered mental status. Some patients showed nonspecific neurological symptoms such as headache, dizziness, and confusion. Generally, after 2 weeks of incubations period, COVID-19 patient will experience a cytokine storm in an attempt to clear the infectious agent. Cytokine storm is a concern for pregnant women as the imbalance of cytokine expressions may expose the fetus to a higher chance of having ASD. Various risk factors can disrupt fetal development due to the fact that neurological development is the product of a multifactorial process that depends on the interaction between genetic and environmental aspects. Infectious agents that can cross the placental barrier and enter the fetal bloodstream can cause direct damage by promoting cytotoxic effect, mitotic inhibition, or any events that lead to vascular disruption. An infectious agent could also induce an aggressive reparative response that lead to increased lesional area and intracranial calcifications. It is known that infectious agents such as rubella, cytomegalovirus, and herpes viruses can alter brain development and cause impairments such as brain calcifications, microcephaly, and other neurodevelopment disorders. Altogether, compiled evidence suggests that these complications reported during pregnancy are associated with neurodevelopment disorders, particularly ASD. Nonetheless, reported studies indicated that the inflammatory process is a common key factor in response to the infections. Better understanding of the normal neurodevelopment and its comparison with neuropathogenesis in emerging viral infection is required for the identification of virus target cells and its possible neurodevelopmental damages. Further researches are warranted to elucidate the precise mechanisms of how immune activation may cause neurobiological and neurochemical abnormalities relevant to ASD. It is also important to evaluate the long-term effect of maternal viral infection and its correlation with ASD. Maternal immune activation involving cytokine network dysfunction may be the potential pathogenic process that links viral infections with the risk of ASD in children. Human cytomegalovirus (CMV) is the most complex member of the human herpes virus family. This virus belongs to the neurotropic beta herpes virus family and can infect various cell types including epithelial cells, endothelial cells, smooth muscle cells, neurocytes, and sustentacular cells of the CNS. The virus enters via the epithelium of the upper alimentary, respiratory, or genitourinary tracts, and excreted in the body fluids such as urine, saliva, tears, semen, milk, and cervical secretion for months to years, and the virus can appear following primary infection, reinfection, or reactivation. Infants can be infected following transmission from their mothers via the placenta, during delivery, or by breastfeeding. Ingestion of genital secretion or breast milk of infected maternal is the main perinatal route of infection. CMV can invade the CNS during any stage of development and maturity of the nervous system, resulting in congenital or perinatal infection. The invasion of CMV into the CNS can cause neurodevelopmental disorders or other neurologic diseases through the interference of neural stem cells proliferation and differentiation. Unlike influenza, CMV infection are asymptomatic and do not result in hospitalization, but despite being subclinical, this virus still induces an immune response that could lead to the occurrence of neurodevelopmental disorder. In addition, CMV can be reactivated throughout lifetime of the host. This occurs when the latent virus switches to a lytic phase of replication when triggered by a combination of external and/or internal cellular stimuli. Not much is understood on how the reactivation of the virus could affect maternal immune system and what consequences they might have on fetal neurodevelopment and ASD. Congenital CMV infection was suggested by an antibody response to the virus, positive virus culture from the urine, impaired hearing, and inflammation of the retina. Prenatal or congenital CMV infection is one of the leading causes of sensorineural hearing loss and other neurological disabilities. Earlier case studies have also reported that children with congenital CMV infection manifested typical autistic characteristics, including failure to develop good interpersonal relationship, poor eye contact, delayed use of language and non-thematic use of objects. There were several cases of severely disabled children with autism and congenital CMV infection. In another study, positive serum CMV-specific IgM antibodies and also CMV DNA in the urine were also observed in children that developed typical autistic disorder. The virus or the ensuing immune response could disrupt the development of brain region or structures leading to the syndrome of autism. Influenza is an infectious respiratory disease caused by influenza A and influenza B viruses. Both influenza A and influenza B viruses are enveloped negative-sense orthomyxoviridae RNA viruses with a genome that contains 8 genomic segments. The host response to influenza virus could offer protection by limiting viral infection within the lung. These factors include alveolar epithelial cells, cells of the immune system. These viral and host factors promote the generation of inflammation in the lung, which attributes to severe influenza-like illness. Like other common viral pathogens such as rubella and cytomegalovirus, seasonal and pandemic influenza infections during pregnancy have also been associated with adverse neurodevelopmental outcomes. Several evidences have linked maternal influenza virus infection to ASD. There have been several papers providing evidence of inflammation in the gastrointestinal tract of at least a subset of ASD children. These findings include immune cell infiltrates present in the colon, ileum and duodenum, as well as increased T-cell activation in the intestinal mucosa. These inflammatory changes are associated with autoimmune responses that could contribute to the observations of decreased mucosal integrity, or leaky gut. The possibility of a connection between peripheral immune abnormalities and altered behavior in ASD is fascinating at this point. On the one hand, it could be that genetic susceptibility or an environmental insult biases the subject toward both brain and immune dysregulation simultaneously, and that these symptoms occur independently. For example, several ASD candidate genes are known to regulate both brain and immune system development and/or function. On the other hand, it is possible that immune irregularities, such as in peripheral immune cells or the GI tract, interact with an abnormal brain to exacerbate behavioral symptoms. Excessive metal exposure has detrimental effect on the nervous system. The developing nervous system is particularly vulnerable to heavy metal toxicity compared to adults, hence metals, which are one of the main environmental toxicants, have been linked to abnormal neurodevelopment. Such metals include lead, mercury, arsenic, cadmium, aluminium, manganese, nickel, amongst others. Environmental toxicants disrupt the process of neurodevelopment either in combination with inherited vulnerability or by epigenetics. A close link exists between the level of environmental toxicants and the increased number of ASD diagnoses. A report showed that exposure to diesel, mercury, lead, methylene chloride, manganese and a wide-ranging assessment of metals during perinatal period is closely linked with ASD. In addition, research an increased blood level of mercury and lead and a notable decline in selenium in kids diagnosed with ASD when compared to healthy children. Furthermore, low level of glutathione GSH has been recorded in ASD patients as a major basis of this disorder. Several neurotoxic metals have been reported to alter GSH production. GSH is an antioxidant in the brain and acts as the major defense system against free radicals produced by toxic metals. Further support for immune system dysregulation theory comes from epidemiologic studies of ASD. It was found that some autoimmune diseases such as rheumatoid arthritis, celiac disease and type 1 diabetes are more common in mothers of ASD children than in mothers of typically developing children. These results and those from prior studies could mean that an abnormal immune system is epigenetically passed on to the offspring. It is also possible that maternal autoimmune reactions could have deleterious effects on fetal brain development. Experimental in vivo and in vitro studies of autism suggested changes in neuroprotein levels, altered gene expression, and neurobehavioral abnormalities after exposure to certain pesticides. The latter hypothesis receives support from the finding that 12 of mothers with children with ASD offspring have anti-fetal brain antibodies in their serum, a figure significantly higher than in mothers of typical children. Animal studies also support the idea that such antibodies could be relevant for pathophysiology of autism. As per MIT understanding, it may be possible that we can effectively prevent the birth of autistic children by regular administration of appropriate nosodes and homeopathic drugs selected on the basis of physical generals and mentals to mothers right from the early months of pregnancy. Molecular imprints contained in appropriately selected homeopathic drugs and potentized nosodes can deactivate the exogenous and endogenous pathogenic molecules and antibodies that act as factors affecting the genetic expression processes associated with the development of neurons and synapses in infants, thereby preventing autism. This knowledge should be widely propagated and practiced as part of routine homeopathic pregnancy management protocol by homeopathic physicians. Homeopathic prescriptions for ASD should be based on totality of symptoms, combined with a causative as well as miasmatic approach, with realization that miasms are nothing but chronic disease dispositions caused by off-target actions of antibodies generated in the body against alien proteins such as infectious agents, vaccines, etc. Symptoms in autistic child varies widely, and should be carefully recorded by direct observations by the physician, and talking to the close family members and caretakers. Symptoms noted during earlier stages of disease development should be carefully collected. History of motional disturbances, infectious diseases, vaccinations and drugs to which mother has been exposed to during pregnancy have to be considered. Peculiar gestures are most important in deciding the prescription. Emotional symptoms, symptoms associated with sleep positions and patterns, eating habits and peculiar cravings, abnormal fears and forebodings, thermal responses etc. also are important. Homeopathic drugs selected as similimum on the basis of these peculiar symptoms should be included in prescriptions. Potentized forms of casein and gluten can play a big role in the MIT prescriptions for autism, since casein, gluten and some opioid peptides present in a range of food sources, are all implicated in the causation of autism spectrum disorders. In autism spectrum disorders, majority of children have wheat and milk protein intolerance. Immunoglobulins such as IgG, IgM and IgA were detected against 9 specific neuron-specific antigens in the sera of children with autism. These antibodies were found to bind with different central nervous system molecules that have sequence homologies to a milk protein called casein. Since many infectious agents including measles, rubella virus and cytomegalovirus vaccines have long been found as etiologic factors in autism, MIT protocol for autism treatment should contain potentized forms of these nosodes, so as to provide the molecular imprints of concerned viral proteins. As such, morbillium 30 and herpes vaccine 30 should be important constituents of MIT prescriptions for autism. Congenital hearing loss is a problem in many autistic children. In such cases, including acid fluoricum 30 in the prescription is found to be helpful, even though its mechanism of action is not explained. This drug was tried successfully on the basis of rare peculiar symptom found in such children. Hearing is better by bending head backwards, which is also found in the homeopathic proving of that drug. Since several evidences gathered from medical records have linked maternal influenza virus infection to ASD, and the population-based cohort study reported almost a 2-fold increased risk of autism after gestational influenza virus infections, it is obvious that antibodies generated against influenza virus play a miasmatic role in autism. Our MIT prescriptions for autism should inevitably contain the homeopathic nosode influenzinum 30. Molecular imprints of metallic elements should be included in the homeopathic prescriptions, if there is any history of pregnant mothers having exposed to heavy metal toxicity from environments. In some cases of autism, heavy metal involvement will be evident from symptomatology itself. Molecular imprints of antibodies formed in maternal body against diverse types of infections and alien proteins also have to be included in this list, since maternal antibodies play an important role in the causation of autism. You can never deal with autism by single drug single dose approach of classical homeopathy, as causative factors as well as involved targets are multiple. Multiple drugs will be required, depending upon symptoms, miasms and causative factors. Do not hesitate to use any number of drugs if indicated, and to repeat them frequently for long periods. Use only potencies 12C or above. Never use low potencies and mother tinctures. According to my view, 30C is the ideal potency. MIT protocol suggests to add all indicated drugs together and dispense as a single preparation and repeat twice daily in drops doses for long period.